Bear with me here. I have to take roll for financial aid. And I should have taken it this morning, but we didn't take a break, so I didn't take it then. And so I'm taking it now. And I'm also remembering who isn't here yet, who I know was there this morning. Mom, is that your phone?
No, that was mine. Sorry.
Oh, you startled me. I was like, that sounds like my, no, it's not my phone. Bear with me. I'm taking roll. I'm taking,
Sorry, I have to get into another class right now, but
all right.
I can stay on for a minute. Oops,
Okay.
hi.
You're all right.
Let me just finish.
I'm taking roll because, and I honestly, I don't take it very often. Like I'll notice if I don't see you for several days or several times that we meet, but I won't notice otherwise. But I have to take it right now because financial aid requires that you show up for class at least one time. And if you aren't in class, you will be dropped from your class. Let's
go outside. Ruby.
bear with me just for a second here. And I know not everyone is here that was here this morning, but yeah, but I know that the majority are. How can I go with that right now? So I want to finish the blood chapter. Oh, let me say something first. Remember that every time we have lab, you have lab exercise questions that are due. And they're through the Wiley lab book. If you didn't, we had a lot of students this week who didn't get them done. And I understand, first off, you don't see the deadline on D2L. Know that they're always due Thursday night at midnight, the following Thursday night at midnight. So we're doing lab exercise 31 today. Those questions are going to be due next Thursday at midnight. Anybody who I can see on the grades does not have us grade right now for it. I'm just going to give you an extension until, let's say, Sunday night at midnight.
Will it be under the chapter 18? Like, is it under that?
Who's talking? Claire?
Sorry, Lexi.
Just Lexi, do me can you share your screen really quick and I'll show you where it is.
Yeah, absolutely.
Bear with me just for a second because I don't think it's going to let you share just yet.
Okay.
Professor,
Yeah,
please, I'm seeing you updated lab virtual lab blood typing now, and I've already done it.
well, I can look and see. I Toyin, I can honestly tell you that I can't specifically, I don't remember specifically what your grade was. Give me 1 second and look at it, but let me show Lexi first where this is. So
Okay,
just a
because I'm
sec.
seeing what I'm seeing now, because I know I did it, so I don't understand what I'm seeing.
Give
That's
me,
why.
I can look, I just can't look right now, so give me 1 second here.
Okay.
Oh, Mary, stop doing that.
Let me know when I can do the share.
You can do it now. I'm sorry, I had
Okay.
to make it, I
It's
had
okay.
to, I have to push a button so you can actually do it. You can do it now.
All right, so I'm sharing the D2L. So if I go into the blood,
So go into the Wiley lab exercises.
okay, so
So scroll down, keep scrolling down. No, scroll down on where the contents, where it says syllabus, class Zoom meeting, scroll down. Don't click on that. Scroll down. Another thing, let's do it this way. Click that content browser. Let me see what that does. Click content browser. You see the down little caret symbol
Yeah,
right
I
next
see
to, yeah,
this
click
one.
on that little, scroll down here. Actually, you know, I'm sorry, Lexi. I am a little brain dead today. Click on content up at the very top.
the
Scroll up.
table of contents.
Nope,
or-
scroll up. Click on content right there. Now scroll down.
Okay.
Keep scrolling. Keep scrolling. You see where it says Wiley lab exercises? Click on that. Now you scroll down. Scroll down. Scroll down. Right here are the virtual labs.
Okay.
Right here, the very bottom is the Wiley lab exercise questions. So
Okay.
if you haven't gotten them done yet, Lexi, do you know if you were able to do them? I don't know.
I didn't even, honestly, I was just going off of chapter 18. I did that and I didn't do this one.
Tell,
Yeah, it was
I
confusing.
go ahead.
But no, but like when I was in there the other day, I looked at like grades and I was like, I wonder if I have to do that blood thing. And then I was like, well, maybe that's just a different chapter. I probably should have reached out to you and
It's all right. Do me a
Yeah, I was
favor.
confused too. I was like, wait, we didn't really go over this part yet. And it was like
We-
put for next week. So
We did in lab. We did the majority in lab.
Mhm.
And what these labs are is we're not doing a lot of hands-on stuff. Do you notice that when you're taking an online class?
Yeah.
One of the perks to these virtual labs is they try to simulate that for you.
Okay.
That's the reason they're being assigned, is because it's the next best thing to a hands-on thing.
All right. Well, this makes much more sense. Thank you so much for showing me
Okay.
where it was at.
You're
'cause
welcome.
I had no idea. To be honest.
Do me a favor. Go up to content again, or go to the course home, either course home or content. Go to course home. I'm sorry, go to course home. Now go to scroll over to the left side there. Not the left side, the right side. I apologize, the right side. You see where it says upcoming events, or even I thought it said right here, click calendar really quick. You see where it's, click the arrow next to it. I'm not helping here. Click the arrow next, go back. Sorry, this is my head. But so click that little thing and say go to calendar. Click go to calendar. Now scroll down. Okay, scroll down a little bit more.
It's not letting me scroll down anymore.
You can't scroll down any further.
Nope.
Shoot. I thought it told you the due dates here.
It is there though, the due dates, as you can
Where?
see, like the 27th, the 3rd, if she clicks on there, it will tell her.
Oh, because of the dots.
Yeah.
Oh shoot.
Okay. So if you click the dots here, it tells you what's due. The only things that aren't here, do you no, look over on this on the right side there. Do you see where the blue dots are under the days?
Yeah.
If you click on one of the blue dots,
Okay. I did.
and it's supposed to tell her when it's due, scroll down.
It doesn't tell you what items though. It's just it gives you a blue dot, but it doesn't tell you what. Like I looked on the all day, like throughout all the time, it's not showing me anything.
It has it at the very
Professor, under
beginning.
each assignment, it tells the due date.
Mhm.
On each assignment, it does tell the due date.
Yeah.
But people are missing them, so I'm trying to figure out. I thought there was another place on D2L where it told you the due dates.
Mine looks different than hers.
Yeah, mine's on my home screen. All of my the due dates for it.
Mine as well.
She has hers somehow set up a little differently, I think.
Mine looks like hers, and if she would just scroll the whole way down to where, because they're due at 11:59, if she would over on the time thing, go the whole way down to 11 at the very end of the day.
Oh, I see it over
Yeah.
here. It's just like
Oh.
cut off.
You know
And-
what I want
we
you to do? Can I ask you to do something? Will you call Hack IT Tech and help them figure out how to get your due dates on your front screen on D2L?
Yeah, I can do that.
Because the only ones that aren't going to show up there are the Wiley.
Okay.
And the Wileys are always due the Thursday night. We do them on Friday. Like today.
I got all of those from the syllabus.
Oh, well, you know, Kevin, you got them, but just keep in mind all the time, they are due, if we do them on Friday, they are due the following Thursday at midnight. So today we're doing, so last week we did lab exercise 26. The questions for that on Wiley that are under the Wiley lab exercise 26 are due next Thursday at midnight. The questions that we're going to do today for lab exercise 31 are due next Thursday at midnight.
Yeah.
I'm trying to keep things a little organized. I know some professors have due dates all over the place, and that doesn't work very well. I'm trying to keep things fairly organized, and I'm sorry, I wish Wiley showed up here, and it just doesn't. Can I have my questions extended? Yes. If you did not get them done, for especially this first lab exercise, because there were a lot of people who didn't get them done, and I understand why. If you did not get them done, I'm going to give you an extension until Sunday at midnight.
Thank
And
you.
I'm gonna do it, you're welcome. I'm gonna do it right after class. I haven't had a chance to do it because with Wiley, I have to go in and manually give every single person that I'm giving an extension.
Okay.
I can't give more than one people the same extension. I have to do it individually. So just be patient with me as I do it. That's what I'm gonna do, Jade. Yes.
Sorry. Do you still want an email or are you going to do it for the
Nope.
entire class?
Don't send me an email because I'm just, I saw who didn't do it. I got them. All right. Jade, can we talk about it after class? Is that all right?
Professor, you
That's fine.
haven't answered me. This is Toyin.
Yep,
I said I did the blood typing, but you didn't grade me. I was
Toyin,
not
I
given a
don't,
grade.
it should automatically put your score in. I don't have to grade it. I don't know why it didn't transfer over. I can look,
But it's showing 0%.
I know, but I can't, I don't know why it didn't transfer over into D2L. I
That's what
have
I
to
had
go,
problem.
I have to go in and look. And sometimes it wasn't submitted and there's a submit button. I don't know, but I can call.
I submitted it and it shows me the grade.
Toyin, I understand, but I don't know why the grade didn't transfer over to D2L. Do you understand what I'm saying? I believe that you did it.
Yes.
I just don't know why it's not transferring over. And so be patient with me and give me a second after class to look and see if I can figure out why it didn't transfer over. And if I need to, I can call McGraw-Hill because it's not like I don't have their phone number on my contacts list. So yes, I will figure it out. And I understand, I understand sometimes that grades do not transfer over. The online stuff is great up until it doesn't work. And then it's not so great. So be patient with me and I'll figure it out. So I want to finish the blood chapter because the last part of this chapter, the white blood cells are all about the immune system. So be patient with me and let me finish this out. Oh, I wanted to readdress the question that Martin asked earlier. Two of his questions from earlier. I think he's here. I saw his name, yes. Martin, you asked about the reticulo. And I made the comment that I thought that reticular tissue meant a net or a filtering. It does. I looked it up. The meaning of the word reticulo is a net or a filter. So you find reticular connective tissue in anywhere where you're going to get filtering. And when we talk about filtering, we're talking about the immune system.
Okay,
so you're going to see it in this. So when the reticulocytes, your question about why they were called reticulocytes, they're called that because they form in the reticular tissue. Does that make sense?
Yeah.
And then I wanted to address I wanted to look at one more thing that you asked about. And the reason I'm going to do this when I feel like one of you does not understand something, what that most likely means is that that person, in addition to a bunch of other people, also don't understand. So I want to readdress something really quickly that we went over earlier. Because I like when you're willing to admit that you don't understand what I'm saying, because what it means is that other people are not understanding also. And I need to know that kind of stuff. Let me scroll down here. So looking here, this question, the blood types are the glycolipid tags are the antigens. Antigens are almost always something that an immune response can be generated against, something that's recognized as being foreign. So these are antigens. The blood type tags are antigens that are made of glycolipids. Glyco meaning a carbohydrate. Anytime that you see this little 6-ring structure right here, anytime you see a 6-ring structure, that's it can be a branchable. Bear with me, I'm sorry, I live right next to a road that the ambulances come down. Anytime you see these 6-ring structures and a branch of them, it's a carbohydrate. It's a sugar tag. So in the case of this, because it's a carbohydrate attached to the plasma membrane, it's a glycolipid because it's attached to the lipids of the plasma membrane. Did that help a little bit? Maybe. Martin, do you think that helped?
Yeah.
Okay.
Miss Mary, I'm the type of learner that I have to keep reading it and going over it a whole bunch of times.
I understand. I understand more than I can say. But the only reason that this answer was not correct, D was not correct, was because the blood type, the blood group is not it's only the antigen, it's not the antibody. If it had been if it had been antigen instead of antigen antibody complexes, if it had just been an antigen, there would have been 2 correct answers here. It would have been glycolipids and antigens. I hope that helped. So I want to finish this out because the white blood cells, if you look at them, they are the mobile units of our body's immune defense. And since our next chapter is immunity and our lab is immunity, it was important to finish this. So we have 5 basic types. We have neutrophils, eosinophils, basophils, monocytes, and lymphocytes. They're the best mnemonic that I know for remembering them in their relative percentages within the normal body is never neutrophils, let lymphocytes, monkeys eat bananas. What that means is that neutrophils are the most common. Lymphocytes are the second most common. Monocytes are the third most common. Eosinophils are the 4th most common. And basophils are the 5th most common. So these leukocytes, leuko meaning that they're clear, like a white substance there that's clear. So white blood cells and leukocytes, same thing, synonymous. Just like we call erythrocytes because urethra means red. Leuko means clear or white. So the immune system is made up of leukocytes, their derivatives, and a variety of plasma proteins. They recognize and destroy or neutralize materials within the body that are foreign to the normal cell. Anything that isn't you is possibly going to activate the immune system. This is why allergies that shouldn't be dangerous are considered foreign for some of us and stimulate an allergic response. Even though they're not toxic to us, our body thinks that they are and works to get rid of them. So the main functions of the leukocytes is that they defend against invading pathogens. They identify and destroy cancer cells that arise in the body. And they function as a cleanup crew that removes worn out cells and tissue debris. Like I said before, their leuco means clear, so these are colorless. They lack hemoglobin. They can vary in structure and function and number. They're somewhat larger than erythrocytes. Erythrocytes are really quite tiny. They're like what, 7.5 micrometers across in width and 2.5 microliters in height. They're not very big. There's 5 different types of circulating leukocytes. There's neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Within the body, remember that mnemonic, never let monkeys eat bananas. It's the easiest mnemonic that I could ever think of. So neutrophils never being the most common, if you look here at the numbers underneath, 60 to 70%. So most common, never. Neutrophils let lymphocytes, which are 25 to 33%. Monkeys, monocytes, which are 2 to 6%. Eat eosinophils, 1 to 4%. And basophils, 25%, 0.25% to 0.5%. If they have granules in them, they are granulocytes. And you'll see that they have, with a polymorphonuclear, is because the nucleus looks kind of odd in there. It's not a really circular or even a U-shaped nucleus. It's like a two-parted, bilobed or several lobed nucleus. So we have polymorphonuclear granulocytes, and we have mononuclear agranulocytes because they don't have granules. Leukocytes ultimately originated from the same undifferentiated multipotent stem cells. Remember, that's what we talked about at the beginning today in lecture, the hematopoietic stem cells. That's where they all came from. Multipotent meaning that they can go in multiple directions with regards to the way they're going to differentiate. So granulocytes and monocytes are the ones that develop in the bone marrow, and they come from the myeloid stem cells. If you look back on that chart that we saw twice this morning, they come from the myeloid stem cells with the erythrocytes and the platelets. Most new lymphocytes, oh, and the other ones, the bear with me here. The lymphocytes are coming from the lymphoid stem cells. So most new lymphocytes are actually produced by lymphocytes that are already in the lymphoid tissues. What this means is that we have lymphocytes that are circulating throughout your entire body. And when they're activated, they come in contact with something that causes them to become activated, which we'll talk a lot about later. They multiply. And the areas where they meet these things that cause them to get activated are in the lymph nodes and the tonsils. I told you about reading that book, Immune by Philip Detmer. He talks about the lymph nodes and the tonsils as being the dating centers where people get activated. I thought it was a pretty good way to think about it. So, or the dating sites, not dating centers, dating sites. So the total number of white blood cells and the percentage of each type can vary considerably to meet the changing defense needs of the body. So neutrophils are our phagocytic specialists. What does it mean if it's phagocytic? What does it mean to be a phagocyte?
It it eats stuff. Phagocytosis is it like eats or like destroys, kills.
They eat things. And one thing that we'll talk about here in a little bit is that they eat things, they break it up, and then they wear it on their outer part of their cell. And I told you I really like this book, this Immune book. If you like science and you want to read a book that's actually makes it kind of funny, read that book, because he talks about the fact that these phagocytes, they eat all this material and then they just smear. It's like eating all these things and then smearing the guts of everything that you ate all over the outside of your body. Kind of a yucky depiction, but you won't forget it. So as your phagocytic neutrophils are phagocytic specialists, and they release this web of extracellular fibers called neutrophil traps or nets that contain bacteria killing chemicals. They see things that look foreign, bacteria, and they kill them. They're non-specific. They're the first defenders on the scene of a bacterial invasion. They're very important in the inflammation response, and they scavenge up the debris, clean things up. Eosinophils are involved with allergic conditions such as asthma and hay fever. And they fight internal parasites. They can attach to worms and secrete substances, those granules in them, and kill them. Basophils are least numerous. So these, remember, never let monkeys eat bananas. So last one. Basophils are the least numerous and the most poorly understood. They're quite similar to mast cells that are involved in promoting inflammation. They degranulate and put out a bunch of molecules like histamine and heparin that stimulate and promote inflammation. So basophils release histamine, which is important in allergic reactions. If you look at almost any of your anti-allergy medications, it's an antihistamine. And they also release heparin, which speeds up the removal of fat particles from the blood after a fatty meal, and also can prevent clotting of the blood. Why do you think you would want the blood to be less likely to clot during an inflammation? Well, let's say someone in here steps on a nail later today. Unless you've been immunized lately against tetanus, you probably want to go and get a tetanus booster because tetanus is a nasty one. However, now you've got an injury in your foot, and it's not just the bacteria that cause tetanus. It's a break in your first line of defense, your skin, one of the first lines of defense, your skin. And now you've got all this bacteria rushing into your foot. Why would it be important to have heparin be a bunch of heparin to explode into the bloodstream and cause it to be easier, be less likely to clot?
Is it so that the white blood cells can be transported faster to combat the-
Exactly. So the white blood cells can move faster. You know, they can get to that area where they need to be faster. And they can move out of the capillaries faster. Monocytes are like neutrophils in a lot of ways. In one major way, they're also phagocytic cells. But they emerge from the bone marrow while still immature, and they circulate for a day or two before settling down in various tissues. And they continue to mature and enlarge in those tissues, becoming macrophages. We've heard that term macrophages before. So big cells, large eaters. I will tell you what pus is, P-U-S. We've all seen it before. Dead macrophages that have eaten up their fill of foreign stuff and now they're dead. There's probably some living ones in there too when you clean out a pussy wound. But a lot of it is dead macrophages. So monocytes become macrophages, and these are professional phagocytes. This is what they do, is they go in and they engulf bad things. Lymphocytes provide the immune defense. These are specifically, we're going to talk about the fact that we have an innate immune system and we have an adaptive immune system. Adaptive meaning that it adapts to be specifically programmed. Innate means that it's non-specific and it's always there. It doesn't have to adapt. It's always there. There's drawbacks and they work together. But lymphocytes provide the immune defense against targets for which they're specifically programmed, meaning they're involved in the adaptive immune system. They're specifically programmed against specific pathogens or bacteria or bad things. They live for about 100 to 300 days, depending on whether they're actively screening. Some of them will live for years and years, and maybe your whole entire lifetime. If you were immunized against measles, mumps, and rubella, that MMR, vaccination, you have cells in your body that are circulating around looking for those antigens that are associated with those disorders. And so they can live about 100 to 300 years, or they can live for your entire lifetime. There's two different types. There's the active ones and there's the memory ones. The memory ones live for can live for forever, for your entire lifetime. The active ones are 100 to 300 days. So B lymphocytes produce the antibodies which circulate in the blood. They're responsible for the antibody mediated or the humoral immunity. Humoral meaning, I want to say humoral means bloodborne, but think of humoral as being the antibody mediated. They're responsible for antibody, that's obviously a duplication here, but they're responsible for the antibody mediated immunity. T lymphocytes, they don't produce antibodies. So why do we need them? We're going to talk a lot about why they're so important. We talked about AIDS for a little while this morning. That is what's attacked with AIDS, is a specific T lymphocyte that's vital. And we'll talk about it when we go into lab, when we talk about lab today. And that's why HIV and AIDS are so nasty, is because they're attacking your immune system. T lymphocytes do not, and for another reason, we'll go into it in a little bit. T lymphocytes do not produce antibodies. They directly destroy specific targets by releasing chemicals that punch holes into the victim cell. And they also work to enhance and mediate other B lymphocytes. They also are one of your major players against regular, bringing down the inflammation once the threat is gone. So they specifically are also involved in targeting cells, including cells invaded, body cells invaded by viruses and cancer cells. In the time between now and this morning, probably several times, your T lymphocytes have killed multiple, possibly multiple cells that could have developed into tumors. So lymphocytes have also, because I told you that they're around for 100 to 300 days, but some live for forever because they have a key function in immune memory. So we saw this kind of before, but here we see the erythrocytes and the platelets also. So here's just an idea about the total count numbers of normal blood cellular formed elements. Which of the following, now that this is, I know this is going back to last week, but which of the following is not contained in the Buffy coat? What's the Buffy coat part of?
The Buffy coat is part of the plasma.
Nope.
Or is it the
Where did we see it? Do you remember, Claire? Where did we see it?
Well, it's between, it's like when you separate blood, it's in between like the blood and the plasma.
You're right. You're right. It's there. What is not contained in it on this list?
E.
Say it one more time.
E.
D is in Delta, or E is
an elephant.
E is an elephant. You're right.
Am I the only one?
Actually, no, wait. You're not right. You're not right. What else could maybe is not there?
I can't see all of them. Is it just my computer? Or
Maybe,
do you need to? Okay,
maybe.
there we go.
Can you see them now?
Not all of them. Yeah, now I can. Okay, sorry.
No, it's all right.
I would say E.
C, because I'm hearing impaired. C is in cat.
Is an egg.
See, that's what I said too.
It's not E. It's not E. What else? What is?
C. It's C, I think, because it has.
Yeah,
like,
Let's
C.
see. Why is it C?
Honestly, I just
Kevin,
picked
why
the
is
one
it
that
C?
nobody else has really picked up.
Yeah,
Why-
those
I
are
just
the
picked the
red
one
blood
nobody else
cells,
really
and they're
picked up.
all at the bottom. They're heavier.
Right. That's exactly right. So you guys, when you're looking at I'm just gonna bear with me here right now.
Took a minute. Jeez.
Oh, we gotta keep life busy. Here, let's see here. So we're looking at this cell like this. We have this capillary tube, it's full of blood. We spin it down. Right here, erythrocytes. So the hematocrit is the percentage of the length of, the length of the capillary tube that's filled with erythrocytes, packed erythrocytes, divided by the total length of the whole blood sample. This is the buffy coat right here. Buffy coat contains the platelets and the lymphocytes. No, sorry, platelets and white blood cells. And it's just a small amount, a layer right here.
So if platelets are dead red blood cells.
They're not dead. They're not dead. They're fragments of megakaryocytes.
Oh.
They're vital because they're the ones that are involved in promoting that platelet plug. That's the first step in hemostasis and stopping blood loss. That is why thrombocytopenia, which is like anemia, but it's a loss of platelets instead of erythrocytes, is so bad. What are the least abundant of the formed elements here?
B.
B is in boy,
Basophils.
Kevin.
Yes.
I can tell you, if you've ever thought about it, how similar B and D sound. T and D, they sound really similar. So thank you. What are the most abundant of the agranulocytes? Remember, these are the ones that don't have granules in them.
Lymphocytes.
Yeah.
You're right. No, wait. Least, most abundant. You're right. I'm sorry. Martin. I'm sorry. You're right. You're right. And I'm getting myself messed up. Welcome to my reality. But here's our chart again. Just to summarize. So all of the formed elements, except for the lymphocytes, are coming from the myeloid stem cells. The lymphocytes and the natural killer cells are coming from the lymphoid stem cell line. And mast cells are part of this myeloid, and they're similar in function. Basophils have a similar function to mast cells. Okay, here's some true or false questions for you. Hemostasis is the production of formed elements in a blood. And Kevin, you can't answer. You guys, what's hemostasis?
Double checking.
I thought hemostasis was the process of your blood clotting.
Right. So and you're right. So what is this, true or false, Martin?
It would be false.
You're right. Blood viscosity stems mainly from electrolytes and monomers dissolved in the plasma.
False.
Why, Becky?
Well, the blood viscosity is the it's like the thickness of the plasma
Of the blood.
of the red blood cells.
Yes. So it has to do with the number of the red blood cells, not the electrolytes and monomers in the plasma.
Right.
All right. Lymphoid hemopoiesis occurs mainly in the bone marrow.
I would say true.
It's true. Oxygen and carbon dioxide bind to, you know what? I don't think I like this one, this lymphoid hemopoiesis. Let's go with true for right now. But yeah, I wouldn't ask that one. Oxygen and carbon dioxide, I thought about it. I'm not going to go into it, but I'll tell you there's a way that I could make it false. Oxygen, carbon dioxide, and carbon dioxide bind to different parts of the hemoglobin molecule.
I want to say false. I want to say false.
Why do you say that?
I thought they attached or bound to the same parts.
I think you're thinking about carbon monoxide.
That would be it. We discussed the carbon monoxide poisoning. So yeah, I was thinking that.
We will talk a lot in when we talk about the respiratory system and the urinary system and acid-base electrolyte balance. We will talk a lot about the fact that carbon dioxide is an acid. I'll show you I could show you an experiment after class, or I'll show it to you then if you don't see it after class. But explaining how the carbon dioxide that's coming out of your mouth can bind with water and form an acid. And it's the same way in your body. So most of the carbon dioxide in your blood travels as dissolved in the plasma as an acid buffer. The liver stores excess iron in as ferritin, not in as ferritin.
I think it's false.
Why do you think it's false, Martin?
Because I thought the kidney stores the excess iron.
It's one of the places where I think it. I don't think, I'm trying to remember, but I think it can get sent to the kidneys, but I don't think it's stored there.
I want to say it's true.
It's true.
Because I have I know it's true because I produce too much red blood cells, and one of the issues is it all will end up in, a lot of it could get stored in my liver and cause a bunch of issues as well.
So that's why you have a high hematocrit, polycythemia.
Yeah.
A person that develops anti-A antibodies only after he's exposed to the A antigen and anti-B antibodies only after he's exposed to the B antigen. True or false? I think there was a question that we had that said that they could spontaneously. There was part in the PowerPoint that said that they could spontaneously form. It's a difference between the Rh antibodies and the Bs. Let me add another question in here. A type O person, let me put it this way, a type O person develops anti-A antibodies only after he's exposed to the A antigen, and anti-B antibodies only after he's exposed to the B antigen. True or false? So a person with type O blood. What do you think?
I thought they always had antibodies.
They can develop them, and that's the thing. They spontaneously they can develop them. Let me add another word to this. This is another question that I hopefully will never put on an exam. A person with type AB blood develops anti-A antibodies only after he's exposed to the antigen A, and anti-B antibodies only after he's exposed to the antigen B. A person with type AB blood. Would you say true or false?
Well, isn't AB the like universal receiver, so they wouldn't create antibodies?
That's a way to look at it. The other way to look at it is he's not going to develop antibodies against himself. And if he's got the A and B antigens, he's not going to develop antibodies against himself. So with the type AB, when I say he's type AB, that means that his body already deems the A antigen and the B antigen as being part of him. So it's not going to develop antibodies against those. Does that make sense? It only develops antibodies when it's deemed as something that's foreign, and therefore could be potentially harmful. That's the end of that chapter on blood. So this is the end. Let's go into the PowerPoint on the lab. What this means is that we finished chapter 18. And I wanted to hit on something because with what Kevin said earlier, and welcome to the way my head works sometimes, and especially when I'm doing what I'm doing right now. I don't normally try to survive on 3 hours of sleep. So bear with me here for today. But looking at the syllabus, when we were talking about due dates, especially with regards to the lab, I didn't put them in here because we tend to stay up to date with labs. We don't tend to stay up to date with lecture. I'm going to try harder to do it this semester. But I also love the fact that we get some amazing dialogue in this class. So scrolling down here, the due dates for the lab, like Kevin said here, they are right here. It tells you what virtual labs were assigned and when they are due. So another place where they can be found is right here in the syllabus.
Thank
I
you
wanna-
again for extending that. I try never to miss an assignment, so I
Don't,
appreciate
it, you know what, it happens. And I don't, you're welcome, because I know it happens. So don't worry about it. So what this also means is that the chapter quiz for chapter 1, and I think they were due the next February 2nd, maybe, are due next week, the quiz and the smart book assignment.
Is there only one smart book assignment? Because in grades it says there's 2. So
It does for the it says that there's 2 chapter 18 smart book assignments.
Yeah, in the grades. And I went back in and I was like working through it for like an hour.
Wow.
And I had already done it. And it was like, oh, I was like, I think it's repeating. And it was like, you already completed this assignment. And so I was
All right,
was
let me
reading
go
for
in
a
and
whole
look. Let me go in and look. But yes, so the chapter 18 smart book assignment and the chapter 18 quiz are due on February 3rd. But let me bear with me just really quickly. Let me look. Or it usually tells me right here whether I posted it twice. It is posted twice, you're right. Here, delete grades. Actually, you know what, before I do this, I will work to remember that later. Who was that, Claire, who said that it was on there twice?
Yes.
Claire?
Yes. Can you hear me?
Yeah. Would you email me and tell me that if I don't remember right off after class, and I'll take it off?
Sure.
Because I don't want to delete it because it's not a good idea just yet. I'm going to look at it, but let's get into lab. And I will tell you that because I have all of you for both lab and lecture. We may do some lecture in lab. Let's just see how things go. I don't want to get significantly behind, but right now our labs seem to be taking up the whole time anyway. So the lymphatic system and immunity.
And you said these lab exercises aren't on Brightspace?
Yep. Let me here, just 2 seconds. It's right here. So this one actually says Wiley underneath it. I should always have them say Wiley underneath it, but it's right here.
Okay, thank you.
Of course. Okay, so why lab exercise terms to define. Do you remember what interstitial fluid was? I'm trying to remember if we ever really talked about it last semester or not. If you had me for lab, we might have talked about it, but I can't remember if we talked about it if you just had me for lecture. And I don't know about the others. Do you know what interstitial fluid is, somebody?
And that fluid between like the cells?
It is, it's a fluid in between the cells. So we have intracellular fluid and we have extracellular fluid. Intracellular meaning in between the cells. We have extracellular fluid that surrounds the cells. And then we have fluid that's in the blood vessels themselves. And I'm, rather than trying to say this, I'm going to do it a little differently. So we have intracellular fluid, which is the fluid inside the cells. Fluid inside cells. We have extracellular fluid, which is the fluid outside of the cells. And the fluid outside of the cells, because it's all fluid outside of the cells. The fluid outside of the cells includes both the plasma, the blood, or actually, let's not say the blood, let's say the plasma, because that's the fluid part of the blood. The plasma and the interstitial fluid. So intracellular fluid, fluid inside the cells, extracellular fluid, fluid outside of the cells. The plasma, or excuse me, the extracellular fluid includes the plasma and the interstitial fluid. So plasma is a fluid within the vessels. An interstitial fluid is the fluid that is in between the cells and outside of the blood vessels. So it's the fluid that's in between the cells, but that's not in the vessels. So the plasma is a fluid within the vessels. Interstitial fluid is a fluid in between the cells. With that in mind, plasma is part of the blood. The interstitial fluid is the plasma that leaks out of the vessels during circulation of the blood. Because that's the fluid that's going to enter and surround the cells. The lymph is the fluid that's going to be taken up by the lymphatic vessels and system. And once it's in the lymphatic vessels, it's called lymph. So it's the interstitial fluid that's taken up by the lymphatics. So once it's in the lymphatic vessels, we call it lymph. Is that clear? So the major functions of the lymphatic system are that it filters the lymph. It also returns the lymph, because remember, the lymph is interstitial fluid that was in the plasma that got secreted out of, that left the capillaries, went and it surrounded the cells, and then it's taken back up into the lymphatic system. So we have to return that lymph to the blood where it was originally, or we're just going to be losing blood volume. So it filters the lymph and it returns it to the blood in the circulatory system. It also is involved in producing and training the immune system lymphocytes, and absorption and transport of lipids and lipid-soluble vitamins. Any vitamin that is lipid-soluble, vitamin A, vitamin K, vitamin D, are all absorbed this way also from the small intestines. So this is a special function of the lymphatic system that doesn't have to do with the immune system or lymph, but it specifically has to do with absorbing lipids from the small intestines. And it does it via what are called lacteals. So the lymphatic system, they are closed, the lymph vessels are closed end tubes. If you look here, you can see it right here. Oh, maybe not right there. You can see it here and right here. They're closed end. So they're not they're not like vessels where they start in one, they flow in one end and they flow to the other end. And they just keep flowing. These are closed off tubes, closed off vessels. They're closed off vessels. And so they only, rather than being able to move both directions possibly, they only move one direction. So the major reason is just to absorb that interstitial fluid and return it to the blood. That's how it works. And of course, as it does that, it's going in, it's going to travel through the lymph nodes, it's going to travel through the thymus, it's going to travel through the spleen, it's going to travel through the liver. And that's also where the filtering is taking place. But so here we have a network of lymphatic vessels or lymphatic capillaries that surround the blood capillaries. And fluid that leaves out of the capillaries and isn't reabsorbed back in ends up being interstitial fluid that's then taken up by the lymphatic capillaries. Your book actually gives a number, and I think it's like 33% or something to that effect of the interstitial fluid is taken up by the lymph system and returned. So we have lymphatic capillaries that merge to become lymphatic vessels with valves. Valves are crucial here because we don't have a lot of pressure. So the valves prevent things from backflowing. So we have lymphatic collecting vessels with valves. We have nodes. And then we have lymph vessels that become lymph trunks, that become lymph ducts to return to the venous system. Questions so far? So lymph vessels are these one-way vessels that surround capillary beds with valves to prevent the backflow of lymph. The pressure in the interstitial area, I'm going to zoom in on this just a little bit. The pressure of the fluid in between the cells here pushes on the overlapping of the cells that make up the lymphatic capillaries. And that is the way the fluid seeps into the lymphatic vessel. You can see that this is a closed end tube. There's little anchoring filaments to help keep things open, keep that vessel open. But it's thin endothelium that's just kind of overlapped, and then the pressure helps things to secrete in. It pushes on this, which allows it to come in. Does that mean, is this clear so far? Doing all right so far? Maybe.
Yes.
Okay. So ultimately, remember that one of the main functions of the lymphatic system ultimately is to return the lymph to the blood. And excuse me, it returns via two ducts. It returns via the right lymphatic duct and the thoracic duct. If you look here, this is where your right lymphatic duct is, right here. Here's your, about right there. Here, or about right here is your right your left lymphatic duct, which is also your thoracic duct. They're named the same thing. So the right lymphatic duct drains this part of the body, and all the lymph that's returned from this part of the body drains into the right lymphatic duct. The rest of the body all drains into the thoracic duct, which can also be called the left lymphatic duct. So here's my right lymphatic duct. Here's my thoracic duct, which, looking at the location, it could also be the left lymphatic duct. We have lymph nodes up in our neck, the cervical lymph nodes. We have thoracic lymph nodes. Thoracic lymph nodes. We have axillary lymph nodes. Remember, axillary meaning your armpit area. We have inguinal lymph nodes. And we have intestinal nodes. Bear with me here. Cisternoculi is right here, right below, right above the kidneys, below the spleen in level. And it is the beginning of the thoracic duct. It's going to go up like this. One other thing that we see here is we see what are called Peyer's patches, which are found in the intestines. They're more specifically found in the, I want to say they're found only in the small intestines. Why do you think we would need to have a mucosal layer in the small intestines? And it's usually part where they're located is like right next to where the small intestines is going to drain into the large intestines. Why do you think we would have a major reason for having a lymphatic tissue right there? What do you know that's stored, that you have a lot in your gut, which is when we talk about your gut, we're talking about your large intestines.
Bacteria?
Yeah, you have a lot of bacteria. If it leaves your large intestines, we work to keep it there. There's a symbiotic relationship of the bacteria, and we have bacteria other places. We have a lot of bacteria in your mouth, a lot of bacteria in your mouth, a lot of bacteria in your esophagus. Why would it be so important to have these Peyer's patches in the small intestines if we know that the large intestines has a lot of bacteria in it? Those Peyer's patches are preventing any possible spreads. One of their main functions is to prevent any small possible spreads of the bacteria from your large intestines into your small intestines, because from there it can go anywhere. That's one of the major reasons I can think of why we would have those specifically in our small intestines, is mainly to keep a check on what's going on in our large intestines. Our stomach is a pretty amazing immune system in itself because its pH is so low. It kills most things anyway. So it's not because of our stomach. It's because of what can come from our large intestines. So major sets of nodes and kind of a review about the drainage. Cervical nodes, axillary nodes. You also have mammillary nodes, inguinal nodes, intestinal nodes. The cisternoculi is the beginning of the thoracic duct, which would be analogous to a left lymphatic duct, that they both drain into the subclavian veins. And we haven't talked about the venous system yet, but what does sub mean? If I say subclavian, bear with me.
Below?
Below, or what else could it be? You're right.
Below the clavicle?
Could it also mean underneath? Probably. So the subclavian vein runs underneath the clavicle. In a few weeks, we'll go through the vessels and we'll talk specifically about those. But that's the reason. So remember, excuse me, that the right lymphatic duct and the thoracic duct are both draining into the subclavian veins. And that's the way it's returning the lymph back to the circulate blood. So right jugular vein, right thoracic duct or thoracic duct, draining both into the subclavian veins. The cisternoculi coming up, becoming the thoracic duct. Major lymphatic organs and tissues. We have two primary ones. We have the thymus, which is a site for T cell development. And we have the bone marrow, which is a site for B cell development. All other organs of the lymphatic system are secondary. So the skin is a secondary organ. The tonsils, the lymph nodes, the MALT, meaning the mucosa associated lymphoid tissue, the spleen, all of these are secondary organs. The only primary ones are the thymus and the bone marrow, because they're the site of T cell and B cell development. So this is a good summary photo. I'm gonna zoom out just a little bit. So red bone marrow is a primary tissue. In adults, it's diffusely located in the central portions of flat bones, with smaller amounts found in the epiphyses of some of your larger long bones. Its function is to act as a hematopoietic tissue. If you look here, look at that just for a second here. And this is no more dogging on your former lab professor. I think sometimes I was telling my mom, I said, sometimes I wonder if they think I'm that good because he just was kind of lousy. But the hemopoietic tissue, if you look at that, is spelled differently than the way it was in our textbook. Do you see that? It's spelled differently and it's pronounced differently. But they mean the same thing. So the main function of the red bone marrow is that it acts as a major hematopoietic tissue, producing all the formed elements of the blood, including the erythrocytes and the leukocytes and the platelets. The thymus is a small butterfly-shaped gland on the superior surface of the heart that degenerates with age. It's really important when you're little because it's of its training of your T cells, but it degenerates over time. So it plays a key role in the programming of the immune system to recognize itself and provide a site for maturation of T lymphocytes. Another one, the spleen, is the secondary largest lymphoid organ. It has a lumbar structure, lobular, and is located on the left-hand side of the body next to the 9th and 11th ribs, between the 9th and 11th ribs. Helps trap foreign material and remove damaged and aged erythrocytes, while also acting as a reservoir for erythrocytes, leukocytes, and platelets. The lymph nodes are also secondary. So they're small bean-like organs found in major clusters. Their primary job is to filter lymph, trap foreign material such as bacterial and viral particles. If you remember, this is, and I told you I really like this book that I read. The spleen and the lymph node are the dating sites for where B cells and T cells will become activated. So the thymus, and I told you I really like this book, so you're gonna have to put up with some of the quotes from this book. He said that this is the place, if you think that your university life is hard or your college life is hard, this is the university of death. This is where T cells are trained not to attack the body itself. If they attack the body itself, there's no negotiation, they're just killed. So this is the training center for the T lymphocytes. So this is an area, it's crucial when you're little, and then it degenerates over time. So it's got 2 lobes. If you look here, it's got trabeculae, a cortex, and a medulla. The cortex, you remember we talked about the different parts of the bone, and we also we kind of talked about it. What's the outer surface of the brain called? What's the outer surface of the brain called? Don't think too hard about this. What's the external part of the cerebrum?
The cortex?
Yeah, it's the cortex. So anytime that you see the outer surface in a histological slide like this is the cortex, the outer part. The medulla, just like the medullary cavity of the bone, is the inner part. In here, we also have these little division points. These are called trabeculae. Trabeculae means little bars. So these are just little separation points. So within these lobules of the thymus, we have T lymphocytes, macrophages. We have dendritic cells, which are specialized phagocytic cell. It's different from the dendrites of the nervous system. I like to get them confused, and they're not. These are immune cells. So T cells, macrophages, dendritic cells. In the bone marrow, remember that we have those megakaryocytes that are putting out platelets. We have sinusoidal areas with reticular cells, and like Martin helped us remember this morning, these are developing red blood cells, the reticulocytes. The spleen is similar. It has, and I'm sorry, this picture is a little blurry. Here's a histological section. We have red pulp, which is blood infused, and we have white pulp. We have trabeculae, those little bars, those little dividing points, and then we have a capsule around the whole entire outer surface. looking on the external surface, we have a hilum, which is where the veins and arteries and nerves are coming in and out. We'll see a hilum. Actually, no, we don't see one, but we'll see a hilum on the kidney, means the same thing. We'll see the hilums on the lungs. It's the same thing. It's where the veins and arteries are going in and out of the organ. It's one direct location. I told you I have a bunch of organs that are in the freezer, and I was telling my mom, I said the one thing that probably would have made this lab, because if I thought about it, I would have asked him for it, is to get a spleen. And I'm sure he would have given it to me if I just asked him for it. But they look really, really, really red. They're dark purple because of all the blood that's filtering through them. They're redder than the liver. They're more purple and redder than the liver. So lymph nodes have an outer cortex, have a medulla, have germinal centers where B lymphocytes and T lymphocytes are interacting and becoming activated and proliferating. We have a subscapular sinus, this area right here, and trabeculae. We have afferent lymphatic vessels and efferent lymphatic vessels. Guys, if you had me last semester, or if you knew this already, what does afferent mean?
Is it like up or?
You're
Oh,
close.
Away,
Say it one more time.
away.
You're in the
Away.
opposite. They're coming in.
Oh, they bring a
Afferent.
take away.
Afferent means incoming. Efferent means outgoing. So efferent, outgoing. Afferent meaning incoming. So it's the same thing here. As we saw it when we were looking at the nervous system, we see it here also. When we were looking at the nervous system, which Gina hasn't gone over, but we're going to go over it in 121. The afferent nervous system. Let's see, now I'm getting myself messed up. The afferent lymphatic vessels. Fluid coming in, efferent fluid going out. We have lots of afferent lymphatic vessels and only 2 efferent vessels going out. Also have valves to help maintain so that things only move one direction. They only move in and out here. Looking at a cross section of the cortex. And germinal centers. These are germinal centers. This is all reticular connective tissue. So we have reticular fibers, reticular cells. Questions so far? Another picture. This is looking at a dissected lymph node on a cadaver. So this is a cross section through it. Looking at, you see the trabeculae, the little bars, the cortex, the medullary sinus, germinal centers, where the proliferation of the B and T cells is occurring. one last, one last part. The tonsils. Used to pull them out of every child that ever had any reason to pull them out, they were pulled out. Now we're realizing they're actually pretty important to the immune system. We have palatine, pharyngeal tonsils, your pharynx up here. So your pharyngeal tonsil right here. Your palate, your top palate of your mouth. Right at the very end, posterior end of your top palate of your palate is your palatine tonsil. Your lingual tonsil, lingual meaning tongue, your lingual tonsil is right at the posterior end, bottom of your tongue. So we have two types of immune responses. We have an innate, non-specific immune system. This is a defense system with which you were born. It protects you against all antigens. It's innate and non-specific. So it involves barriers that keep harmful materials from entering your body. I told you I really liked this book, and it was really fun to listen to it, and it even helped me connect some dots with the immune system. He talks about the skin as being a major hellhole because it's got a high acid to it, and it's barren, and there's very little ways to penetrate it. And so very few things actually survive on it. That's why it's such a great barrier until it's penetrated. So so it's one of your first lines of defense. This innate, it's part of your innate immune system. This barrier that forms the first line. Your adaptive or your specific immune system is your receptor-mediated immunity. Receptor meaning receptors bind specific things. And it always involves some form of contact. It's specific. With the assistance from helper T cells, B cells can differentiate into plasma B cells that can produce antibodies against specific antigens. The humoral immune system deals with freely circulating antigens from pathogens or outside of infected cells. So differences between your innate and adaptive immune system. In your innate immune system, this immunity takes effect very early, right after exposure to infection. So within hours to days. Your adaptive immune system is delayed. It's days to weeks. It has to adapt. It's got to get ready. It has to get prepped. So your innate immune system helps significantly and is essentially holding things at bay until your adaptive immune system is geared up to go. So your innate immune system activated by a wide range of pathogens, non-specific. The adaptive immune system is specifically activated by certain components. of pathogens, certain antigens on pathogens. Whether it's an antigen on a red blood cell or an antigen on a pathogen, if it's foreign, it's foreign. The innate immunity confers broad protection because it's non-specific. Adaptive immunity is what is involved with why vaccines are effective, giving your body a chance to prepare. Primary functions of the innate immunity is that it controls the spread of infection and directs the development of the adaptive immunity. Primary functions of the adaptive immune system is that it's clearing infection and the development of a memory response. Questions so far? Comments? Guys were much more vocal this morning. I'm wondering if you're getting kind of tired.
Yeah, very tired.
Yeah, very tired.
And it's snowing where I live. So the weather's.
Yeah.
I
No,
live
listen,
I always have more energy in the morning.
you know what, and I am a night owl, so I don't tend to have high energy. I was really glad it was this class this morning because I was really worried how I was going to make it through this morning. My mother can tell you. So differences between the cells and components of the innate versus adaptive immunity. In the innate, remember this is non-specific. So dendritic cells, remember these are like they're phagocytic cells. Macrophages, phagocytic cells. Natural killer cells, they just kill things. My mother's smiling because she probably remembers what the guy in the book said. Killer cells. It is funny. I'll tell you in one moment. We have to wait until we get there, and then I'll tell you. Basophils are, remember, these are your leukocytes, your granulocytes. So basophils, eosinophils, neutrophils. They're all part of this innate immune system. It doesn't matter what kind of parasitic worm it is, the eosinophils are going to try to kill it. The complement system, we'll talk about that here in a moment, but it facilitates in a lot of ways. It facilitates and moves things forward. In the adaptive immunity, we have B cells and T cells. These are specific. They have receptors that require that they only specifically bind things. Things that can overlap are some T cells and natural killer T cells. So we have three major barriers in our defenses. We have a non-specific physical and chemical defenses. So these barriers, examples are skin. Like I told you in this book, they calls it a hellhole. Very few things survive on your skin. The mucous membranes, can you imagine trying to survive in the slime that runs up and down your nose? Things just don't survive very well there. That's why it's such a great defense. It's also full of macrophages. And it's slimy and it's slippery and things get stuck in it. The acid in your stomach has a pH of 1 or 2. Very few things are going to survive that adventure. You have lysozyme, or when your tears, your lacrimation, lacrimation. Lysozymes in tears. When you expel things, coughing, sneezing, vomiting, diarrhea, this is also involved in these defenses, getting rid of what's trapped there. The next part of your innate immune system, still innate, so non-specific. Macrophages, neutrophils, eosinophils, basophils, and monocytes. Remember that macrophages are developed from monocytes, which are one of those leukocytes. So this could say monocytes, neutrophils, eosinophils, basophils. Actually, wait, that doesn't work. Monocytes become macrophages. Just go with that for right now. It looked like a good figure until I read it just now. The complement system is also here. We'll talk about how it's involved in enhancing immunity. Adaptive immunity is your cellular. So this is your T cells. Humoral meaning your B cells. These are receptors, they're specific. It has to adapt. There's a delay in it getting up to snuff to do what it's supposed to. So different parts of the non-specific innate, lysozymes and tears. You have natural flora. Even though I told you that we keep those Peyer's patches there in your small intestines to keep the bacteria in your large intestines in check and keep it from spreading, it is good at keeping out, because by its very existence, it prevents other bacteria from being able to grow on in that area. One thing that I learned about your skin is that I've told you I'm an animal person, you know that. And I learned, I volunteered with the Humane Society for years in Idaho. And while I was there, I dealt with ringworm in kittens. And can I tell you how bad, nasty that stuff is? But one thing that I learned with ringworm, it's a fungus. It's on the skin. And if you go on an antibiotic, or you put your cats on an antibiotic at the same time that they're struggling with ringworm, guess what happens to the ringworm? It goes insane. It starts taking over a lot more skin, because the normal bacteria on your skin, the normal flora, that's what flora means here, bacteria, is keeping that kind of stuff in check. And so when you put someone on like a cat on a whole body wide range systemic antibiotic, it kills the bad bacteria that you're trying to get rid of, but it also kills a lot of the good bacteria, including the bacteria on the skin. So this normal flora, this normal bacteria, this is another part of our immune system that's innate, and that keeps things in check. So mucous membranes, tears in your eyes, your saliva, Also has a lot of lysozymes in it. Blood proteins, your lungs, your stomach, normal flora, and the mucus tract coating of your urinary tract. Any questions about the barriers involved in the innate immunity? One thing that is a pretty amazing cascade, and it's not like the clotting cascade that we saw earlier, but it's still a pretty big cascade, is what we call the complement cascade. And it's involved in augmenting inflammatory processes. Inflammation is an innate, non-specific immune defense. The complement cascade involves in working as a chemotaxin. What is a chemotaxin? A chemotaxin is a signal that helps other cells to move towards it. So let me give you an example. Later today, you got that example earlier about stepping on a nail. You go out, you step on a nail. You've had your tetanus shot, so you're not overly concerned. But inside all your immune cells in your foot they're going in, that life is not good for them. All of a sudden there's an emergency. They're sending out signals telling other immune cells to get down to your foot. Those signals are your chemotaxins. Chemotaxis means to move towards a signal. So chemotaxins are the signals that you're moving towards. Opsonants are things that coat bacteria, making them look better to other cells that will kill them. So this opsonin right here, it actually means to make more tasty. So it coats pathogens, making them look more desirable and helping the immune system to think that they look more yummy to eat. So you're coating them to make them just look better and entice the immune system just a little more. Just stimulate the immune system just a little more. Kind of like putting icing on a piece of cake if you really like cake icing. Just making it look a little better. The complement system also promotes vasodilation. Remember, like Martin said earlier about why we use heparin, get blood flowing to an area. Well, we also want to promote vasodilation and vascular permeability. We want the vessels to enlarge, and we want the cells, especially the leukocytes, to be able to leave the vessels, but by increasing the permeability of the vessels, the leukocytes can leave the vessels and move out into the area of infection. The complement system also stimulates the release of histamine from the mast cells and activates kinins. Kinins are inflammatory signals. So the complement pathway can be initiated in 3 basic ways. We have a pathway where it's activated by the classical pathway, activated by the classical pathway, meaning antibody antigen complexes. The antibody binds to an antigen, and the complement pathway sees it and is initiated by it. So these proteins here of the complement pathway see these antibody antigen complexes and activate this protein C3 to become C3A. Lectin is a protein that binds to sugars on microbes, on their plasma membranes. And it can work to activate this complement pathway. The alternative pathway is C3 or CB that enhance inflammation by binding to and interacting with pathogens directly or apoptotic tissue. Apoptosis meaning cell tissue that's dying. So all three of these different ways can initiate the activation of C3A to become C3B. C3A can continue on to develop more enhanced inflammation.
Did you say sorry.
Go ahead.
Did you say apoptotic means dying tissue?
It mean, yeah, it's programmed cell death.
Okay, thank you.
So it helps you to be able to destroy tissue that's already dying.
Okay, thank you.
So C3A can also go on once it's activated and it breaks away from C3B, and C3B goes on in its own way doing other things. C3A enhances inflammation. C3B works in that opsonization. That's to make something, coats a pathogen, making it look more desirable to cells that are going to destroy something. So you coat the bacteria with C3B, and all of a sudden the natural killer cells or the macrophages think, oh, that looks a lot more tasty than it did a second ago. Let's eat it now. That's what opsonization means. So C3B coats bacterial things or antibody antigen complexes and makes them look tastier. C3B can also go on and convert C5 into C5A and C5B. C5A is involved in enhancing inflammation. C5B signals the MAC, initiates MAC, which is membrane attack complex. You guys get the basics of this down and recognize that we're going to go over it in lecture next week also. Get the basics down, but recognize that we are going to go over it again too. So the membrane attack complex, terminal pathway, it is essentially what it does is it activates a bunch of proteins that make pores into cells, foreign cells, pathogenic cells, and lyses those cells. That's what MAC means here. And that's what is initiated by C5b. The C here always means complement. You see that a lot here, C, 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 C. That's always complement. So 3 different pathways initiate this complement path cascade. Antibody antigen complexes, lectin that binds to sugars on bacterial cells, or pathogens, or an apoptotic tissue that by itself activates C3b. Questions about this? Get that general idea and just recognize that it's working to help inflammation and enhance innate immunity. And the major player that starts a lot of this is C3a. The major complement proteins that you between now and next week should remember is C3a and C5. So non-specific, different structures and defense mechanisms of non-specific immunity. The skin, the physical barrier to penetration by pathogens. Secretions contain lysozyme. It also has bacteria on it. The digestive tract, high acidity of the stomach, and is protected by the normal bacterial population of the colon. The respiratory tract, secretion of mucus, movement of mucus by cilia, and alveolar macrophages. You have macrophages that live in the lungs to help eliminate pathogens. The genital urinary tract, the acidity of urine and vaginal lactic acid. The sperm actually have to fight with this vaginal lactic acid for fertilization to occur. Internal innate immunity, phagocytic cells ingest and destroy bacteria, cellular debris, denatured proteins, and toxins. Interferons, well, guess what? They interfere with the replication of viruses. Kind of a name all in itself, but that's what they do. Interferons, they interfere with the replication of viruses. Complement proteins promote the destruction of bacteria and enhance the inflammatory response. Endogenous pyrogens. You guys, what's a pyromaniac?
Someone who sets fires or obsessed with fires.
Sets fires, that's right. So what endogenous pyrogens, they're the ones that induce fever. They trick the hypothalamus into thinking that it needs to be elevated to a higher temperature. So these are secreted by leukocytes and other cells, and they produce the fever. Natural killer cells destroy cells infected with viruses, tumor cells, mismatched tissue, transplanted tissue cells. And the last one, mast cells, release histamine and other mediators of inflammation, and cytokines that promote adaptive immunity. Remember that inflammation is a major part of innate immunity. So my mom, now that she's gone to bed, we can talk about the killing of action of the natural killer cells. These are actually, yeah, before I say that, make sure I'm saying it right. But these are cells that essentially are looking to see if there's something wrong with any cell. Remember, these are innate. They're non-specific. And they're just looking to see if there's something wrong. And if there's something wrong, if you look at it, and the book is better about it. You'll have to read the immune book that I read. But he gets really, really close. He looks at it, looks at the enemy cell for a minute. He looks to see if there's something not there that should be there. And if that something is not there, he puts out all these perforants and that form holes in the cell and destroy it. He's looking to see, and they're not specific. Anything that looks like it could be foreign, or looks like, you know, if there's something missing from it, there should be there, it's going to kill it. I like my mom, like he says in the book, he says he's not going to try to ask him what's wrong and try to console him. He's just going to put a bag over his head and shoot him. It's terrible, but you won't forget. He so puts out a bunch of perforants, and through these perforants we get granzymes being sent in, and these granzymes then degrade all of the enemy cell enzymes within the cell. So we get lysine of the cell on top of the fact that we're introducing these granzymes. There are enzymes that then go in and destroy all the cell's enzymes. The enemy cell then dies by apoptosis. And the macrophage engulfs and digests the dying cell. Let's talk specifically about naturally acquired immunity, artificially acquired immunity, naturally acquired immunity, and active acquired natural immunity, active artificially acquired immunity, passive naturally acquired immunity, and artificially acquired. If it's acquired, that means it's adaptive. You have to acquire it. It's not, it's specific. So when we're naturally acquired active, means that you run into something in the wild. It stimulates your immune system. You get some pathogen into your body in your normal adventures in life. You get some pathogen into your body, and your B and T cells then become activated specifically against it. That's naturally acquired. Artificially acquired means that you get a vaccine. You get something that's is a very small amount that shouldn't be harmful to you. And your body gets a chance to respond to a very small amount of an antigen and develop a specific response. So naturally acquired meaning you just run into it. So I'm going to use the COVID vaccine. I'm not going to get political. I apologize if I offended anybody this morning with my political rant. Or my whatever.
Not
Okay.
at all.
Well,
No.
well, if it did offend, I apologize. Naturally acquired, meaning let's say you get COVID and you develop antibodies against it, and eventually, if it doesn't kill you, you'll develop an immunity against it. Artificially acquired means that you get a small amount of an antigen from that virus. We introduce it via a needle, and you develop your B cells and T cells develop a specificity against that. Passive naturally acquired most often happens when, I think the most common is mothers breastfeeding passes antibodies onto babies. So passive meaning they don't have to make those antibodies. They're already there. Sometimes, and they don't tend to live for forever because they don't develop a memory. Active all involves developing an immune memory. Artificially acquired means, bear with me here. Artificially acquired is when, for example, you get a snake bite and it injects a toxin into you, and hopefully you can fight it off before it kills you. And you can develop your adaptive immunity before it kills you. Well, some toxins are so bad that you're not going to develop because adaptive immunity takes a couple weeks or a couple days. You may not survive long enough. So what they do is they give you a serum from something else that developed antibodies against it, like an animal serum that can develop antibodies against that toxin. And they give that to you via an injection to block the effects of that toxin. Is this making sense?
Mm-hmm.
Yes.
Yeah.
Yeah.
And like I said, we're gonna go through this again next. We're gonna talk about it again next week also. Secondary and primary and secondary immune responses. Antibody B cells and T cells that find an antigen that they can bind to, they're specific. So they have receptors and they can only bind to specific things. When they bind to their specific thing, B cells will proliferate. They will proliferate into antibody secreting cells and memory cells. Memory cells are the ones that stay around maybe for your whole entire lifetime. Antibody secreting cells secrete antibodies against whatever the antigen you were exposed to. So for example, let me, 1 second here. Actually, this is probably not the best place to put this slide. I should have thought about where this slide was. I'm going to skip it for a moment and come back to it. So here's our antibodies. Antibodies have a constant region. They have this Y-shaped region. And they have a constant region here. Constant region that's all of this. And then they have an antigen binding site, and it's specific, very specific. You see, we also have complement binding sites because remember the classical pathway for that complement cascade gets activated via antibody antigen complexes. So we have these two binding sites. This is on an antibody, two binding sites that can bind any antigen. Different antibodies based on what is here. This is the part of the antibody which will tell will dictate what happens next after the antigen binds here. Epitopes are part of the antigen that binds to this spot. Not all of the antigen will bind here. Only this part. Only this part. That's the epitope. The whole structure is the antigen. So B lymphocytes that are involved in antibody-mediated immunity. Each lymphocyte has surface receptors. Remember, these are your surface receptors. And wait, let me say one thing really quick before I go here. B, on the surface of every B cell, we have our receptors that look just like this antibody. So we have the B cells floating around, and they have receptors that look just like this. So each lymphocyte has surface receptors for the binding of one particular type of many, many antigens. Upon binding with its matching antigen, the B cells then will proliferate. Bind to this receptor that looks exactly like this. It proliferates. Most B cells are going to differentiate into what are called plasma cells that then go on to secrete lots and lots of antibodies. Other B cells become memory cells. They proliferate and become memory cells that are just like these cells, but they're not secreting antibodies. We have some that are secreting antibodies. We have others that are just memory cells. So they stick around and they float around possibly for the rest of your life and wait for that antigen to come again. And when it comes again, they will be stimulated to proliferate again, just like these up here. They'll proliferate again and become antigen secreting plasma cells and more memory cells. This process of B cells being activated by binding to their antigen, their one specific type of antigen that they've waited their whole life for, is called clonal selection. They only actively divide and proliferate if they bind their antigen. Why does this matter? And I should put this slide down there. This is why we get vaccinations, why we get small vaccinations. We get the first vaccination is what we call an IgM antibody. We'll talk about the different types of antibodies next week. But we get a specific type of antibody. We get that COVID vaccine, and I'm sorry, just put up because it's the only vaccine I can think of right off the top of my head lately. Get the COVID vaccine, we get a small amount of that antigen. we have a B cell somewhere floating around in our body, maybe 3 or 4, that eventually is going to run into that antigen that we just received in that shot. When it runs into it, it's going to bind to it, just like down here. It's going to bind to that antigen, our COVID antigen, and it's going to start to proliferate. And some of them are going to become plasma cells that start secreting antibodies that can all bind to that COVID antigen. And some of them are going to become memory cells that will proliferate, and they won't secrete antibody, but they'll stick around for a very, very long time, waiting to see if we ever run into that COVID antigen again. When we run into it again, those memory cells will bind to that antigen again, and we'll get more proliferation. This will happen all over again. And so we'll end up with even more memory cells than we ended up with the first time when we get that booster. You guys, if this is not completely clear, be patient, because we're gonna do it. We are gonna talk about it again next week. So one thing that we haven't mentioned here that are actually really important is that B cells, and I'm gonna try to make sure we finish on time. B cells require the assistance of T cells to become memory cells and plasma cells. So T helper cells will bind also to the antigen and to the B cell and stimulate B cells to proliferate. One thing is we'll realize, and with B cells, there's lots of levels of activation, because inflammation, when it goes awry, is really, really bad. And so there's 2 things that have to happen for B cells to be activated. They have to be exposed to a macrophage or pathogen. And then they have to be activated by a T helper cell. If the T helper cell is not around, we will not develop memory cells. We'll only develop plasma cells. One thing that you need to keep this, I know this is a question, one of the questions on the Wiley thing, that that B cells are going to differentiate into plasma cells in both the lymph nodes and the spleen. Remember, those are the dating sites where B and T cells come together and decide whether they can work together or not. So how do antibodies help physically hinder invading microbes? They can do it a couple different ways. They can bind to the antigen, and I know this is really, really small. They can bind to the antigen, and by binding to it, if you can imagine, you're this microbe, you're trying to move around, and you have this thing stuck to you, and it's not going away. And it kind of looks like a little bit of a crab if you look at it. And of course, then it sticks to another one of your buddies, and now you and your buddy are stuck together. And each of these little antibodies has 2 places where it can bind. And so eventually you're dragging you and your friends are all stuck together. That's neutralization. If you and your friends are trying to do damage, but you've got all these antibodies stuck to you and they're not letting you go. That's how antibodies can physically block things from being able to do what they want to do. Alternatively, remember that antibodies can activate the complement system. Antibodies can also enhance phagocytosis by acting as opsonins. Remember, these are making things even more tasty to phagocytes. And they can stimulate natural killer cells. This slide is terrible. I thought that would be better writing. I think we kind of already went through it before. Remember how that you have to, the B cell has to be exposed to the antigen. And then it becomes memory cells and plasma cells. Plasma cells secrete antibodies. The memory cells continue around and around until it runs into that antigen again. The one thing that I want to repeat is that in order for this clonal selection to occur and for the memory cells to be produced, we have to have a T helper cell. So T cell receptors have an antigen binding site just like B cells do, and I apologize, where did that come from? They have, on their membrane, they have a receptor almost identical to the T cell receptors. It looks very similar. It's the same antigen. If you look here, here's the B cell. It's got a bound antigen, and the T cell binds to that same antigen. That's how they're activated. That's how the T cell can further activate the B cell. Two main types of T cells. We have CD8 cells, which are cytotoxic killer T cells. And we have CD4 cells, which are helper cells, mostly helper cells. The cytotoxic T cells destroy host cells by harboring anything foreign. The cytotoxic T cells also can release chemicals that destroy targeted cells. CD4 cells modulate the activities of other immune cells, and they secrete chemicals that amplify the activity of other cells. They modulate by amplifying things. The differences in what we call the major histocompatibility complex is what's going to determine whether or not we get a T cell activation, a CD8 cell activation or a CD4 cell activation. So we have these, it's called the major histocompatibility complex. It was first discovered when, was first discovered when during tissue transplants because everyone has their own MHC1 molecule. Every individual has a unique, has a very unique MHC molecule. So this is what holds the antigens on a cell surface. So for a CD8 cell to be activated, it has to interact with a class 1 MHC molecule that's found on the surface of all cells. So these MHC1 molecules with an antigen bound to them are recognized only by the CD8 T cells or the cytotoxic cells. The CD8 cells then links the two cells together. And linked in that way, the cytotoxic T cell can then destroy the body cell. It's activated. So CD8 cells only bind to the MHC molecule, 1 molecules. MHC2 molecules, these are found on all phagocytic cells that then can become antigen presenting cells. Class 1, 2 MHC molecules are found on the surface of immune cells with which helper T cells interact. They're recognized only by helper T cells. So these are CD4. So the CD4 receptor links the two cells together, and the activated helper T cell must bind with the MHC2 molecule to activate the B cell. Helper T cells have to bind, and that's when you will create memory cells. Without the helper T cell to activate the B cell, you will not create memory cells. So this is just a comparison of CD4 and CD8 cells. Remember, CD8 are cytotoxic. CD4 are helper T. Helper T cells, what do? They stimulate innate immunity by activating macrophages, by stimulating that chemotaxis, those signals, and stimulating inflammation. They also work through clonal selection of B cells. They also work to activate clonal selection of cytotoxic T cells. T cells carry out this cell-mediated immunity. They don't secrete things, so they have to be in contact, cell-to-cell contact. They're clonal and antigen specific, and they're trained only in the thymus. They're just like B cells in that some of them are going to go on to be active plasma cells. Others are going to be memory cells. Interleukins, let's just say really easily, interleukin, if you think interleukin works in between leukocytes. So it works to interact and facilitate actions between leukocytes. There's a lot of information there. I'm not expecting you to know it all. I'm just giving it to you, just providing it. So CD4 helper T cells can secrete interleukins that are going to activate T cells, T regulatory cells, B cells, T effector cells, natural killer cells, T helper cells, and monocytes. So think of interleukins as being able to work between cells. T cell activation works the same way with regards to antigens. It gets exposed to the antigen and then it gets stimulated. Some become effector cells that go on to do the action right then. Others become memory. Last part, or almost the last part. T cells become immunocompetent, meaning that they're capable of being having immunofunctions in the thymus. B cells become immunocompetent and mature in the bone marrow. This last slide is just summarizing this material. I am well aware that that's a lot of material. And let me say one more thing, interferons. Interferons are things that are put out by virally infected cells that help protect other cells from becoming infected by viruses. If you think about interferons, they interfere with viral replication. Interferon gamma can also stimulate T cells and macrophages and dendritic cells to be more likely to eat other things. And then this is one more, just a general idea of adaptive versus innate immune system. Guys, I know we're out of time. Do you have questions for me? Looking through this PowerPoint, I made sure last night, I made sure that every one of the questions that you were being asked would be covered by this material. Let me know how it goes.
Okay.
All right, Professor. My question is, do you ask me to wait after the class?
Yeah, because I need to figure out what's going on with you. So figure out what's going on with your stuff.
All right.
I might be able to figure it out right now. So I can try.
Are we good then?
We are good. We're good.
All right. Thanks, everyone. Have a great weekend.
Thank you. Have a good weekend.
And you too. Bye-bye.
Have
Bye.
a good one.
Have a good weekend.
Thank you.
Welcome.
Get some sleep, Mary.
You too. You're the one who's been sick.
I know, and I can't. And this is my second round, and I've had my vaccines.
Oh, this is COVID.
Mm-hmm.
Oh, yucky.
COVID. Yep, second round.
But see, you're not ending up in the hospital. That's
for sure. I have,
as you cough.
Yeah, it could always be worse. The exhaustion's killing me with working